Our second speaker today will be Dr. Carolina Terragna. Dr. Terragna joins us from the University of Bologna in Italy, where she is currently responsible for the research activities and molecular diagnosis of multiple myeloma and acute lymphoblastic leukemia within the Institute of Hematology. Carolina earned her biology degree at the University of Milan, as well as her doctorate degree, studying the use of immune globulin heavy chain rearrangements as a tool for MRD analysis in multiple myeloma. She has written more than 200 publications and articles in international journals and is a leading researcher in the molecular biology of hematological diseases. Today, she will share with us some of her vast experience with respect to MRD analysis in multiple myeloma. Her presentation today is titled Assessment of MRD by NGS in Daily Clinical Management of Multiple Myeloma.
I will talk about the minimal residual disease evaluation by next generation sequencing in the daily clinic management of multiple myeloma. Multiple myeloma is a lymphoproliferative disease affecting plasma cells. Plasma cells is the final differentiation stage of lineage B and are specialized in antibody production. The B cell maturation up to plasma cells require several processes, the most characterizing of which is the rearrangement of the heavy and light chain gene of the immunoglobulin. It is believed that error in this process might be the cause of plasma cell neoplastic transformation by possibly causing aberrant recombination between different chromosomes, leading to translocation and or copy number aberration, which typically characterize myeloma plasma cells. Until the beginning of 2000, myeloma therapy was largely unsuccessful, with very low probability for patients to survive more than 5 years. More recently, the therapeutic landscape of pathology has greatly improved, mainly by the introduction of different classes of very effective drugs. The most effective of one are the monoclonal antibodies, overall targeting the most critical survival mechanisms of plasma cells. The efficacy of these novel drugs combination have improved the median rate of complete response among patients, and consequently the patient survival expectancies, which have increased from a median to 3 to a median to 8 to 10 years for young newly diagnosed patients. However, myeloma still have a fluctuating clinical course with alternating phases of response and to therapy and disease recurrence, with response phases even shorter and recurrent phases more and more aggressive up to a final stage of refractory disease. This dynamics has been recently explained by the clonal evolution theory, which, by applying knowledges of species evolution, ecology, and population genetics, has helped to understand how tumors might respond to selective pressure exerted by therapy. So, two main points are critical to understand the relationships between the biological characteristics of myeloma and the clinical dynamics over time. First of all, the genomic heterogeneity of myeloma plasma cells, which is very high already at diagnosis and which change continuously over time, might explain the evolution dynamics of the disease. Second, the minimal residual disease, the residual cells after therapy, whose quantity and quality correlate with patient's prognosis and might explain the disease outcome of each patient. Today, we will focus on the analysis of minimal residual disease, and this is the outline of my presentation. We will talk about the utility of MRD analysis in multiple myeloma, how can we evaluate minimal residual disease in myeloma, and what is the clinical impact of this type of analysis. Let's start by analyzing the utility of MRD analysis in multiple myeloma. As already mentioned, the rate of good quality response in myeloma has greatly improved over the years, thus highlighting the importance of the depth of response. Overall, remission means reduction of the tumor burden under the limit of detection of the methods employed to detect it. And a very well-known concept is that the higher is the tumor burden reduction, the lower is the probability that the disease recurs. So the response depth is very important for patient outcomes, and the management of myeloma patients require the awareness of how many residual disease persist in the marrow after treatment. In fact, it has been repeatedly demonstrated that the achievement of MRD negativity is one of the most important prognostic factors impacting patients' survival. And lately, this is confirmed by these large meta-analyses, including more than 8,000 patients coming from 25 different studies. And more importantly, this is true in different clinical contexts. For instance, for newly diagnosed patients, transplant eligible or transplant ineligible, for relapsed patients, and also for patients carrying high-risk genomic features. Residual disease results are not only prognostic factors, but might help in prediction the disease recurrence, thus potentially allowing the early clinical intervention aimed at containing the neoplastic clone re-expansion. So several evidence support the utility of MRD analysis in myeloma. And how can we measure MRD in myeloma, and what are the most critical features of MRD testing suitable for MRD monitoring? The most commonly employed methods for MRD analysis in myeloma are summarized in this slide. Neoplastic plasma cells locate mainly in the bone marrow and have a patchy distribution, which might be well appreciated by imaging techniques such as PET-CT or MRI. This technique might be used to stage the disease diagnosis, but also to monitor its distribution over time. Also, residual disease can be studied within the bone marrow, either by analyzing the aberrant phenotype of plasma cells or by employing molecular marker suggesting the presence of residual disease. More recently, the wide distribution of the disease in the body has finally suggested that the disease might spread throughout the peripheral blood, thus highlighting the possibility to use the peripheral blood to monitor the disease. Today, we will focus on molecular biology approach of minimal residual disease within the bone marrow. The overall aim of the molecular biology is to detect disease-related marker. This is an issue in myeloma because the plasma cell is highly heterogeneous from a genomic point of view, and therefore clonality marker need to be employed to monitor the disease. And in particularly, the immunoglobulin heavy and light chain rearrangement has been commonly employed to this aim, even though this marker is patient-specific, thus forcing the search for patient-specific marker. Therefore, this is the reason why, among the different possible molecular approach for MRD evaluation, such as ASO-PCR or digital PCR, next-generation sequencing has been considered the most reliable. First of all, because it facilitates the detection of the clonal patient-specific marker, and also because it is highly specific in the detection of a small amount of rearranged IgH or IgK in responding patients once the rearrangement has been detected at diagnosis. Moreover, next-generation sequencing is a high-throughput technology, thus perfectly matching the need of high sensitivity, which has been shown to be highly meaningful in multiple myeloma, as shown here. In fact, MRD positive patients stratify according to the quantity of residual disease, with patients with a residual disease in the range of 10 to -4 doing significantly worse than patients with a disease with a residual disease in the range of 10 to -5 or 10 to -6. Thus highlighting the importance both to reach high sensitivity and to be able to discriminate between different levels of residual disease. So how can next generation sequencing be implemented in the daily clinical practice? Is there room for this high throughput and expensive technology also outside clinical trials? From a technical point of view, the answer is yes, since most requirements for this technique are met, as we briefly showed. However, there are still lots of debate around this topic. Overall, we can say that MRD negativity is evaluated by an NGS or by flow or by imaging has become a major endpoint in clinical trial. And this slide shows the most recent active clinical trial testing minimal residual disease in a critical point during the therapy of myeloma, that is in induction, during transplant, on the maintenance, and more importantly, are using MRD negativities as clinical endpoint, emphasizing its critical role as compared to complete response as evaluated by conventional parameters, which used to have this role just a few years ago. However, the use of MRD results for the management of myeloma has not been widely accepted by the scientific community. And in fact, it is common to attend scientific session where this topic is debated between those who are and those who are not in favor of the use of MRD in this context. Unfortunately, also several MRD-driven clinical trials have been recently proposed, designed to validate the role of MRD results as assessed in specific time points critical for the management of myeloma. But what happened in the meantime? In the real life, the implementation of MRD in the daily management of myeloma patients mainly relies on the propensity of any given institution toward the translation of clinical knowledges into the clinical practice, as well as on the affordability of this translation. In our institution, we decided a few years ago to set up the minimal residual disease monitoring in patients with multiple myeloma achieving very good clinical response. At that time, we had available at our institution the Thermo Fisher S5 NGS platform, and so most data I'm going to present derives from this platform. And also we decided to employ the LymphoTrack assay for MRD analysis, which allows the analysis of IgH and IgK. In agreement with the clinical team, we decided to include in this type of analysis newly diagnosed patients, transplant eligible, achieving very good partial response, that is a very good quality response. Also, transplant eligible patients carrying high risk feature, and these patients are analyzed between the first and the second auto transplant. Also, we included transplant newly diagnosed patients, transplant ineligible, upfront treated with novel regimens. This type of patients have been monitored once a year. More recently, we also decided to include patients that we enrolled in a study aimed at the comparison of MRD monitoring in the bone marrow and in peripheral blood. And the bone marrow in these patients is collected every 6 months. To date, we included in the workflow 150 patients. And here are listed some of the technical aspects that we have dealt with. In our routine, as I said, we implemented the assay on the Thermo Fisher S5, even though we are now moving to the Illumina MySec. We decided to use AGH-FR1 and IG-kappa as primer sets in first, and only in case of polyclonal results, we then use IGH-FR2 and FR3. We also defined the cutoff in order of minimum acceptable number of sequencing reads in order to define which runs can be considered evaluable and which not. And we finally defined a clone as any rearrangement whose frequency was higher than 2.5% of the total IGH reads or 5% of the total IG-kappa reads, and at least 2 times higher than the third most common clonotype as suggested by the assay that we are employing. Here are summarized as other technical feature. I just would like to underline that for each newly diagnosed patients, we performed CD138 enrichment, and therefore samples were highly enriched in neoplastic cells. We also decided to optimize the workflow by including 12 patients in each run, evaluated by 2 sets of primer, and the median percentage of sequencing reads supporting a clone was 58%, ranging from 3.8 to 94%. Most cases were successfully characterized by FR1 and IG-kappa set of primers, whereas 6% of patients of sample resulted polyclonal, and therefore patients cannot be monitored during follow-up. Finally, we also were able to confirm the bias in VH gene segment usage, as frequently shown also by the other authors. MRD has been measured so far in 50 patients for a sum of 75 samples. Again, some technical feature. In each patient, we perform a quality control of the samples by evaluating its hemodilution in order to exclude from the analysis or bone marrow aspirates who were not representative of the bone marrow tissue. For each analysis, we employ 6 micrograms of DNA in 3 replicates of 2 micrograms each, aiming at a sensitivity of at least 10 to -5. We also use for each run a low positive control, and an internal control has been spiked in every reaction in order to be able to define the clonal frequency of the clonotypic rearrangement, and also in order to be able to compare results coming from different runs. Here are summarized some of the results that we obtained so far. Patients can be stratified in 2 subgroups. In the first one were included patients analyzed every 6 months. These are 17 patients and a total of 27 samples, most of which resulted MRD positive. The second subgroup of patients included 33 patients, mostly analyzed during maintenance. We analyzed a total of 46 samples, most of which resulted MRD negative. Important to note, there is a subgroup of patients that are highlighted in blue here that after lots of internal discussion, we decided to report as positive, not quantifiable, since the sequencing results were positive, but the statistical confidence of the results was not sufficient to consider the quantification reliable. The minimal residual disease in these cases was mostly very low, and these cases were prevalent during maintenance. Finally, thanks to the experience that we have acquired, together with other Italian groups involved in the same topics, we decided to set up a working group, actually split in 2 subgroups, one mainly devoted to molecular biology and the second one mainly devoted to flow cytometry, aimed at the standardization of methods for MRD assessment in myeloma in the daily clinical practice. 5 Italian groups were included in the molecular biology group, that is Bologna, Milan, Turin, Pisa, and Rome, and 10 Italian groups in the flow cytometry groups. Both groups are focused on the harmonization of methods already in use in their own institution, on the standardization of the procedure, of a shared procedure for the assessment of MRD in the daily clinical practice, the definition of specific time points for MRD analysis, and also in the identification of hub centers, possibly locally supporting the regional hospital interested in the analysis of MRD in multiple myeloma. We started the activity a few months ago, and we are now working on the setup of a quality control round, and in particular in the preparation of mock samples for validation experiments. Before concluding, I would like to summarize a few points. First of all, the prognostic role of minimal residual disease in myeloma has been repeatedly demonstrated in several contexts, and the technology to evaluate MRD can be considered available to date. However, the real meaning of residual disease still needs to be fully explained, and several questions still need to be answered before the assay would be fully legitimized to be used in the daily clinical practice. Therefore, to date, even though MRD results might support the clinical decision, they cannot be considered the only driver to these purposes. From a biological point of view, the minimal residual disease remains mostly an unexplored world, even though possibly highly informative to explain the mechanism of resistance that each patient might develop to escape the selective pressure of therapy. Similarly, MRD negative results might hide unexpected dangers, especially reminding that multiple myeloma has a patchy distribution and that false negative results might happen. Moreover, myeloma has a spatial heterogeneity. Therefore, different focal lesions might carry different prognostic factors and therefore might differentially contribute to the disease reappearance. And with that, I would like to conclude by thanking all my collaborators in Bologna, and particularly Marina Martello and Silvia Armuzzi, who have been involved in MRD analysis, and you for your attention.